Well, first, let me congratulate all of you for organizing this event. I'm obviously honored and humbled to be able to contribute however much I will. And I thank everybody for staying up, for those who are in zones where you are up, to listen to this. As you can see on the first slide, there is a contact email for me at the bottom. There's also a drtheoharides.com site, which is fairly simple to navigate, and it has a lot of questions and answers. In fact, many of your questions and answers from today, I will try to also reply and post on that one. And then there is a much more complicated but more detailed site at the end, mastcellmaster.com. I would like to stress that there is a very useful YouTube presentation at the very bottom that was in fact recorded almost 10 years ago in Denmark. It involves two patients, one from Denmark and one from the United States. And we had called it My Mystery Symptoms and Mast Cells because, as I'm sure many of you know, and as it will become apparent today, many of the symptoms are fairly mysterious and very complicated and affect different organs. Now, I have a lot of slides, and many of the slides are busy, not only because I believe you ought to know the evidence, but because you will receive these slides, and therefore you may be able to use them if you talk to your own physicians that might not have had time to read and show them the publications. And many of the publications are not necessarily mine. So you'll not sound like I'm biased. Let me start with some historical and very basic features. First of all, back in 1901, two scientists, Richet and Portier, were doing an experiment at the Oceanographic Museum in Monaco. And they actually grounded up the tentacles of the jellyfish. They injected them into dogs, and they believed the dogs would be protected from subsequent stings. And they had a word ready for it. They were going to call it prophylaxis. Instead, however, when the dogs were re-stung, they dropped the blood pressure, the bronchi closed up, and they died. So they changed the name to anaphylaxis, and we use the word ever since. Very interestingly, without knowing each other, in about, and this is a mistake, 1887, not 1987, Paul Ehrlich was staining various tissues, and some cells in the tissues, when they were stained with a dye called toluidine blue, turned violet. And some of the cells were very compact, as you can see on the left-hand side. Some were much more expanded. He did not know what those cells were, but she thought that they were feeding surrounding cells. And because the word for the breast, which feeds the baby, is mastos in Greek, he called them mast cells or mastocytes. And the name got stuck ever since, even though the mast cells don't feed other cells. Now, if we look at the bottom right, you see a micrograph about 1,000 times magnification. You see a blood vessel looking round, and all the red arrows show these blue cells around the blood vessel, and these are all mast cells. So mast cells are uniquely found around blood vessels in every tissue in the body. I will go to the box at the right-hand side in a second. But we always thought that the major trigger of the mast cell is immunoglobulin E and antigen. But the mast cells are in the tissues, and immunoglobulin E is circulating in the blood. So how do the mast cells see the immunoglobulin E? Well, this excellent paper in the journal Immunity some years back showed a mast cell. This is a still picture from a video, in the middle of a bifurcation of a red blood vessel, and you see it in two different types of optics. And the arrowheads show philopodia that the mast cell throws between the endothelial gaps, and they sense what is happening inside the blood. So the mast cells can sense everything outside the body because they're found in the nose, the eyes, the mouth, the lungs, the skin, the gut, and the brain that I will talk about later. And they sense everything that happens inside the blood vessels. So I and some colleagues believe that the mast cell is the first cell that will see danger and orchestrate an immune response. Now, the mast cells have existed, as you can see in the box, for millions of years, even in species that never get allergic reactions, like fish, insects, lizards. And therefore, the mast cells must play some very useful roles, which may be able to discuss during the Q&A. In mammals, they derive from the bone marrow. They have distinct proteins or characteristics on their surface, and we recognize them. They have specific receptors for immunoglobulin E, and they contain about 1,000 granules. So the mast cell is like a soccer ball filled with about 1,000 little ping balls, and each ping ball has about 100 different molecules inside, and histamine is only one of those. And when the mast cell is triggered, as you will see later, it can release not only the content of the granules, but make even more molecules. So we're talking about possibly 200 molecules being released from the mast cell. And we literally, unfortunately, still deal primarily with histamine and a little bit more tryptase that I will talk about. So this caricature is everything you ever want to know about a mast cell. On the surface, I indicated various receptors, but the receptors, even though they're shown in particular places, they're really scattered throughout the surface of the cell. So if we look at about 11 o'clock, we see receptors for immunoglobulin E, and when these are bridged by an antigen or some other molecule, calcium ions flush inside the cell, and the cell degranulates. It literally explodes within 30 seconds or so. And if you look at the left-hand side at the bottom, this is a mast cell micrograph from our laboratory, about 2,000 times magnified. You see the brown area is the nucleus. The dark small granules are intact granules, and the ones that look larger or more violet are the ones that are releasing their content. Yet, within 24 hours, the mast cell restores its granules and it's ready to fire again. So many of the information that is presented in this slide is actually in this review, which was published in the New England Journal of Medicine almost 10 years ago. And I was very grateful to the journal because it allows us to use in the title, the name mast cells, mastocytosis, about which I will talk about, and related diseases as well. It was the first time that in a journal of that prominence, one would talk about related diseases that we will talk about today. However, in addition to the receptor for immunoglobulin E, about 7 years ago or so, other colleagues identified another receptor shown at about 9 o'clock, MRGPRX2, which is triggered by not allergens or antigens, but by positively charged drugs and peptides. We call it sometimes pseudo-allergy. But as you can see at the left-hand side in the publication, the receptor for IgE communicates with this receptor. And this is very important to keep in mind, and something that many of my colleagues miss, that you can have individuals with allergic histories, sometimes we call it allergic diathesis, and that they're triggered by something else, and now the mast cells respond to everything under the sun. So because such receptors communicate, when one is activated, it can lead to activation to other receptors as well. When the mast cell degranulates, it releases numerous molecules, some of which are shown in the box. And the important ones are histamine, of course. Proteolytic enzymes, I call them meat tenderizers, like chymase and tryptase. But they also release molecules such as platelet activating factor, PAF, which is involved in allergies, but it is very difficult to measure in clinical laboratories, and tumor necrosis factor. Over an hour or so, they may also release molecules such as leukotrienes, which are very bronchoconstrictive, and prostaglandins, which activate sensory nerve endings. If, however, we look at the right-hand side and look at the caricature in the middle, which is also taken from the New England Journal of Medicine, many drugs, peptides, cytokines, environmental triggers can stimulate the mast cell with no allergen, no immunoglobulin E. In fact, if we now look at about 12 o'clock, there are receptors for peptides released from nerve endings that are involved in pain, such as substance P, and corticotropin release hormone, CRH, which is the main hormone released under stress. At about 2 o'clock, you see receptors for molecules released from other immune cells, called cytokines or interleukins. And at about 3 o'clock, you can see receptors for every pathogen, such as bacteria, fungi, viruses. These are called toll-like receptors or TLRs. What is amazing is that when these receptors are activated, it takes about 6 to 24 hours, but the mast cell now releases molecules, all of which are pro-inflammatory, such as interleukin 1, 6, 31, etc., chemokines in the second line, which draw other immune cells in the area, and inflammatory peptides, such as bradykinin, neurotensin, and vascular endothelial growth factor. But quite important for some of our later discussions, the mast cell respond to heavy metals, such as mercury and aluminum, which unfortunately are adjuvants in vaccines, as well as to the most common herbicides, such as atrazine and glyphosate. And as you know, glyphosate, at least in the United States, is sold as a Roundup. Now, with this slide, I'd like to give you the background of all the mast cell-like diseases so that you can at least categorize them in your mind and hopefully explain them a bit. Again, you can relate back to the review in the New England Journal of Medicine, where it was shown that the mast cell being in the middle, depending what the trigger is, the mast cell can release different molecules and it can affect every organ or tissue in the body, including the brain. Now, we separate the mast cell disorders in 3 categories. The primary are what we call mastocytosis, where you have a lot more mast cells, either in the bone marrow, or in your spleen, or in your skin. Then we have the secondary, which is pretty much what everybody knows about: allergies, itching, urticaria, rhinitis, conjunctivitis. And then we have the idiopathic, which are very confusing. And unfortunately, idiopathic in medicine means that we don't really know what is going on. And you might have anaphylaxis, you might have edema, you might have itching. And then we have activation of mast cells, where we don't know what the trigger really is. And I will explain the difference between what is mast cell activation syndrome and mast cell activation unspecified. Let's just jump to the bottom for a second, because we use terms in medicine that are very confusing. We say allergic diseases, well, they involve mast cells. We say atopic disorders. These are allergic diseases, but only immunoglobulin E is the trigger. Then we say mast cell diseases or disorders in general that might be involved in all the categories: primary, secondary, or idiopathic. And then we use terms such as mast cell activation disorders. And as you will see, only one of these categories is called mast cell activation syndrome. And if you now look In the middle panel, you see that there are diagnostic codes which are different for each type of diagnosis. So you might have allergic urticaria, you might have idiopathic urticaria, you may have atopic dermatitis, etc. But the most important thing is we should not necessarily be insisting on mast cell activation syndrome, or MCAS, because there are other diagnostic codes, such as secondary mast cell activation or other mast cell activation disorders. Therefore, we should start with mast cell activation unspecified until certain criteria are actually fulfilled, in which case we might go to mast cell activation syndrome. But at the end of the day, it is mast cell activation that we're interested in and how to deal with it. And we shouldn't, again, be stuck with mast cell activation syndrome because it is only one of many such disorders. Now, there are ways, obviously, to diagnose allergies. And I will go through this, and I will repeat this a little later as well. Firstly, let's look at the color pictures. If I were to see anybody with black circles under their eyes, unless they haven't slept for 3 days, that is telling me that something is happening with the mast cells. And if I scratch the underarm or the back with my nail or some dull object, within a minute or two, we might develop these red-looking lines, and these are called dermatographism. From the Greek, you can write on the skin, and then they disappear in about 2 minutes. That means that the mast cells in the skin are responding just to the pressure. There's no allergen there, just the pressure. That is telling me that the mast cells in this individual are likely to be responding to other things that we haven't quite identified yet. So what I usually ask for is the total amount of immunoglobulin E, but also the subclasses IgG1 and IgG4, because IgG4 first is involved in food intolerance, and food intolerance is very different than food allergy. And because, as you can see in the publication at the very top, they found increased levels of IgG4 in children with autism. And I will return to that a little later. Then I ask for certain immune Ig species against certain allergens, because you have to specify which allergens you're looking for. And I usually ask for alpha-gal, which is found in primarily beef and pork, not in fish or chicken, and not in humans, but it is found in many supplements and drugs and cosmetics that contain mammalian products. And I've seen many people getting red on their face because basically they use chopstick or lipstick that contained alpha-gal. Also try to cover at least gluten, dust mites, fungi, grass, and pollen. And depending on what place in the world you live in, then you might choose the pollens, whether it's cypress trees, or pine, or something else. About 50% of people with chronic itching have an autoimmune problems. The body makes antibodies against the Ig receptor, but instead of the antibodies blocking the receptor, they activate. So sometimes we call it basophil activation test, or mast cell activation test, or anti-Ig receptor antibody test. We can do a food intolerance panel. I like doing such panels. However, many of my colleagues don't, because if you eat something every day, it turns out to be false positive. So I usually ask families, if possible, not to eat anything other than boiled rice and chicken for like 3 days, and then do the test. Then we can measure certain cytokines, prostaglandin, tryptase, and tryptase is an enzyme that is found only in mast cells, even though we still don't know exactly what it does, and we have no way of blocking it. And as of recent, I've been measuring vascular endothelial growth factor, because of this publication shown in the red box, where they measured such factors in the blood and they found them to be high in mastocytosis. I don't tend to measure histamine in the blood because it's broken down within 1 minute and it ends up in the urine. So we measure in 24-hour urine. It absolutely has to be collected cold and sent to the laboratory cold. The breakdown products of histamine, which are methylhistamine or MIA, methylimidazole acetic acid, prostaglandin D2, the metabolite of the prostaglandin D2, and I did not list there now. We also measure leukotrienes in the urine as well. So what are the common conditions that we usually think about mast cell activation? Well, as I said earlier, is the type 2 category, where you get allergic rhinitis, asthma, atopic dermatitis, etc. Then you have the sort of more idiopathic or non-atopic allergies. And as I said, mast cell activation syndrome is the one that most everybody talks about these days. But I want you to notice in the long table at the right-hand side that there are many conditions that involve many organs, and every condition listed there with an asterisk involves mast cell activation. So even though it is not the primary condition, therefore we will not call it mast cell activation disorder or syndrome, there is mast cell activation. And therefore it becomes very confusing because you can present with conditions that involve any one of these organs where there is mast cell activation, and it will be absolutely confusing to the health provider because they don't know what exactly is going on. This is why I urge patients, when they visit a health provider, try to focus on 1 or 2 most important, 3 at the most, symptoms or organs to capture their attention. Get someone to be listening to you, and then maybe you can say, you know what, here's my list of 30 other symptoms. If you just present with 30 symptoms, they're just not going to believe you, or they will just be too confused to deal with you. Now, one very important aspect of the mast cell, as you can imagine, is that it causes itching. But as you will see, it is involved in pain as well. If you look at the green box, you see how many molecules released from mast cells can cause itching. We call them pruritogenic. And as you can see, there are papers that talk about the role of mast cells in itch, and especially most recently about the role of platelet activating factor in itch, as well as a molecule that is called interleukin 31. And in the middle panel, you see a paper of ours showing that many triggers can make mast cells to release interleukin 31. Interleukin 31, which unfortunately it is not measured in clinical medicine as of yet, is much, much more itching than histamine. And therefore, in many patients who do not respond to antihistamines, we have found that interleukin 31 is involved. Now, if you look at the left-hand side, it is the cover of the journal Investigative Dermatology. And it's an amazing cover because the red shows a blood vessel, all the green cells are mast cells, and all the diagonal yellow lines are nerve endings in the skin. And if you look at the paper at the left-hand side at the bottom, they talk about mast cell-specific receptors that are involved in what we call neurogenic inflammation. We will talk about inflammation more in a few minutes. And the caricature shows that damage in any tissue can activate the mast cells and it can activate the sensory nerves. The sensory nerves release substance P, which then triggers the mast cells through this new receptor that I mentioned. And the mast cell release molecules that activate the sensory nerves, and you have a cyclic response. And at the right-hand side at the bottom, I show a paper where elevated levels of this receptor in the blood were found to reflect the severity of patients with chronic spontaneous urticaria. So keep in mind that many of the surface receptors can be shed from the mast cell and can be measured in the blood as well. Because of such findings, we believe now more and more that mast cells are involved in neuroinflammatory conditions, including autism that I will talk about later, about long COVID, about which I have 10 slides after I finish today. If there's time, I can review those as well. Myalgic encephalitis, chronic fatigue syndrome, etc., etc. And I just chose some papers to list on the slide. So again, if you have any of these conditions, you can go to your provider and say, well, there is evidence that in fact the mast cells are involved, such as neuroendocrinology of the mast cells in migraines, in fibromyalgia syndrome, and at the very bottom to the right, you see Neuro-Immune interactions in chronic pain. Let me spend a few minutes talking about mastocytosis and how we think about it. So if someone has a history, as you can see in the box at the top, of reactions to foods, to latex, exercise, stress, hot water in the shower, then all of those make us think that the mast cells may be involved. And on the left-hand side, you can see we can talk about cutaneous or skin mastocytosis or systemic mastocytosis, the most common of which is called indolent. So what do we do? Well, going back to what I showed you a little earlier, we can measure a whole bunch of things in the blood, but primarily we focus on tryptase because it is not released by any other cell type. If tryptase is high and the number has changed, the most recent number now is less than 15 nanograms per ml. It used to be up to 20. But be it as it may, if it is high, then we have to worry what might be going on. So what do? You do an ultrasound of the spleen and the liver. If those are enlarged, then you have a problem. If someone has gastrointestinal problems, one might be able to do a colonoscopy and a biopsy, but that's kind of touch and go because you don't really know where to biopsy. Most colleagues biopsy the ileocecal junction, and usually we say if there are more than 25 mast cells per high power field, that's indicative or suggestive of a problem. Otherwise, you can do flow cytometry on the peripheral blood, and the mast cells in the blood are there as progenitor or precursor cells before they go into the tissues where they mature. If, however, they express on the receptors, these receptors on the surface, CD117, 25, 2, then that's indicative that most likely we're dealing with mastocytosis, in which case you do a bone marrow biopsy. Not a bone marrow aspirate, because in an aspirate you pull, and the shearing forces of pulling will destroy the mast cells. So it have to be a biopsy. Now, if you look at the bottom, if you have islands of about 50 or so mast cells, not just 2 or 3, then clearly you have mastocytosis or systemic mastocytosis. If, however, you have lesions on your skin showing on the right-hand side, and here are many lesions, you can have even a single lesion called solitary mastocytoma. Then you have cutaneous mastocytosis. For many of us, this is pretty obvious, or you can do a biopsy and you find out that you have connection to mast cells. If all of those are negative, then I'm repeating what I said earlier. You go to the urine, collect 24-hour urine, refrigerate it, and you measure the metabolites. And then you end up with mast cell activation syndrome if the tryptase was elevated. The tryptase was elevated, but the urine measurements are high. Then we're talking about mast cell activation unspecified. Now, some years ago, my colleagues, for the first time, identified what they call mast cell activation syndrome, and they had certain criteria. And as you can see in the blue box, the criteria was to have a history of mast cell-related problems in 2 or more organs, measure various things, either in the blood or in the urine and have them elevated, specifically tryptase. But what was music to my ears back then, if you look at the upper left-hand side in the blue box, it said also involving neurologic complaints. Even though the neurologic complaint was part of the diagnosis back then, for some reason, my colleagues have dropped that, even though there's more and more evidence that there are neurologic problems. Dr. Butterfield has published numerous papers showing very convincingly that the metabolites in the urine, especially leukotriene and prostaglandin, are more reflective of the severity of mast cell activation than tryptase. And I wrote an editorial talking about the need to define a subgroup of patients with what I call idiopathic mast cell activation syndrome, or otherwise mast cell activation unspecified, because not everybody has tryptase elevations. And not only that, but you have to have a tryptase elevation within 48 hours of an episode. So unless someone has an indwelling catheter, how are you going to draw blood within 24 hours? And most emergency rooms don't know anything about mast cells and tryptase to even measure it. Now, I want to stress, however, before going on, that there's also something we call histamine intolerance. As I said, histamine comes from mast cells, but as you can see at the right-hand side of this slide, histamine is very rich in many foods, such as especially ripe avocado, cheese, nectarines, sardines, spinach, spices, tuna. So therefore, you might be getting symptoms from histamine either because the mast cells are activating or because you're consuming these foods. And the problem is that for histamine to be broken down, you need a major enzyme called diamine oxidase or histaminase. This is why I usually ask for gene polymorphisms for this enzyme and some other enzymes listed in this box that are involved in breaking down histamine and some other products. This is important because by polymorphism, we mean whether a gene is working properly or not, and you can have either homozygous problems, in which case the gene is not working well, or heterozygous, when it's working sort of okay. And this is important because there are supplements containing diamine oxidase that we can give to patients about 15 minutes before a major meal. Now, why am I stressing this so much? Because a lot of people might have histamine intolerance and not necessarily mast cell activation. It may be confusing. And because a lot of patients, especially children, present with abdominal pain. And this paper from Nature 2 years ago or so was incredibly useful because they showed that food antigens can stimulate mast cells in the intestine, and histamine and possibly other mediators shown in the caricature at the bottom left can then sensitize sensory nerve endings, and you might present with abdominal pain. And in fact, this was so important that the New England Journal of Medicine carried an editorial, as you can see, an allergic basis for abdominal pain. So therefore, when you complain, my colleagues should be listening to you because there's a lot of evidence as to why you will be complaining, because something is actually happening due to mast cells releasing mediators. Now let me switch to a little more science. We talk about the mast cell, as I said, degranulating if you have a truly allergic response, or undergoing maybe release of selective mediators a little later, depending on the triggers. So I'll brag a little bit. I published this paper in the journal Science back in 1978 as a graduate student at Yale University, where we showed for the first time ever, if you look at the right-hand side, at the top, if you take any collection of mast cells at any time and you trigger them, about half of them just don't respond. And the other, as you can see with the black dots, do respond. We used a dye called ruthenium red and we put a complementary filter. That's why the granules look black. So number one, do we know which mast cells do not respond? we don't. In fact, as we speak, we're doing experiments to find out is there something unique about the mast cells that do not respond, so that we might be able to identify it and maybe turn it into treatment. If you now look at the middle panel, published again many years ago, we showed that interleukin-1, which is released from mast cells, can stimulate actually the mast cells to release interleukin-6. And both of those are very pro-inflammatory. You might ask, how does that happen? Well, if you look at the electromicrograph at the right-hand side of the bottom, it's actually a cryo-immunoelectron micrograph from our laboratory. And what you see in the red rectangle is a collection of black dots. Each dot identifies an interleukin-6 molecule. And the reason we can see it is because we have an antibody that recognizes the molecule, and the antibody is stacked with a gold particle, and the electron microscope sees the gold particles as black. So you might say, what is the big deal? We know mast cells release. The big deal is that this collection of interleukin-6 particles are only about 100 nanometers, while the secretory granule of the mast cell is 1,000 nanometers. The size of this is almost identical to the size of the synaptic vesicles that release neurotransmitters in the brain. We will never see this for light microscopy. And this is, we call this differential release, because a mast cell can release different molecules at different times to different triggers without degranulating. And to make things even more complicated, if we look at the left-hand side, this is a paper we published recently about mast cell activation beyond histamine and tryptase. And this caricature is taken from this journal. And as you can see, the mast cell not only degranulates, as you can see about 2 o'clock, but it has many different ways of also releasing molecules, all the way to creating nanotubules that communicate with other cells. And now if we look at the table at the right-hand side, this is a list, which I'm sure it's grown since the publication, of receptors on the surface of the mast cell, more receptors than the ones I showed you earlier. And if you look at the right-hand side at the bottom, a recent paper identified even more receptors. And at the left-hand side, a recent paper identified even more molecules released from mast cell. So there's no end to what the mast cell can do. It can respond to almost 100 different triggers. It can release almost 200 molecules. And we are absolutely silly to be focusing on just histamine and tryptase. Now, how about a molecule that is released from damaged cells? And I apologize, I misspelled alarmin, double A there. It's called interleukin-33. So interleukin-33 has, over the last 5 years or so, been associated with mast cell activation and inflammation, and it actually augments whatever other triggers do. And you will see the evidence. So is this real? Well, let's look at the paper at the upper left-hand side. They're talking about a brain-skin connection where stress, and we'll talk about stress shortly, worsens dermatitis via substance P. So substance P is a molecule that I mentioned earlier that is released from nerve endings. So in the middle is a paper published in the Proceedings of the National Academy of Sciences of the United States, where we added substance P and interleukin-33 together in human cultured mast cells, and we measured release of tumor necrosis factor. And as you can see, we have thousands of units being released over control. This amount is more than any other immune cell I've ever studied. If you look at the bottom, another paper from us, also in the Proceedings of the National Academy of Sciences, where we did the same thing, but now we measure interleukin-1 beta. And as you can see, hundreds of units when we combine the two. In fact, as you can see, the mast cells can release two of the most pro-inflammatory molecules? No, TNF and IL-beta. And as I mentioned earlier, interleukin-6 can be released. And at the top, you see 3 publications from 3 different groups. One was us, indicating that levels of interleukin-6 in the blood reflect the severity of the mastocytosis better than tryptase. Now, you might argue, so why are we not measuring interleukin-6? I do. But my colleagues might say interleukin-6 is not specific, and they're right. But if there's no other disease, there's no other condition going on, and interleukin-6 is high, that might be a way of indicating that the mast cells are firing. And if you look at the bottom from Dr. Metcalf's laboratory at NIH, you can see that if the mast cells have this mutation, which is the most common mutation that allows the mast cells to proliferate, not only they proliferate, by now they release constantly interleukin-6, and interleukin-6 acts back on the mast cells. So you can see that the mast cells are very dynamic cells. Not only they can change their phenotype, their reactivity, depending on the tissue, but depending on the triggers as well. So let's take a deep breath, talk about stress. We know what stress is. It can be psychological, it can be physiological, it can be economic, it can be political, it can be all kinds of things. Well, this is a publication from the Mastocytosis Society in the United States, where they showed that heat and stress were the most important triggers of mast cells. No allergy, just the stress. Now you might say, well, you're stressed, go and see a psychiatrist. That might be a good idea. But as you will see shortly, we know exactly what happens. So it's not just in your brain. We know exactly what happens and how we might be able to address it. And in fact, we and others have published many papers showing the impact of psychological stress on mast cells, as you can see in the middle panel. Now, we're getting a little closer to what I will be talking about shortly. But first, I want to indicate a wonderful paper published in Science about 2 years ago, where they showed what I never expected, and I'm sure many of my colleagues never expected. We always say, or at least we used to say, that the mast cells mature after the baby is born. And in fact, many times we don't do any allergic testing until 2 years, because we say, well, the mast cells are not ready to fire till 2 years. Well, we were proven wrong. They showed both in humans and in animals that the mast cells in utero can respond to anything the mother is exposed to. And that's very important, not only because, as you can see, stressful events during pregnancy can increase the blood IgE, but it can also make the newborn baby to have more or higher risk of developing atopic diseases, atopic dermatitis, etc. So whatever happens during pregnancy can definitely affect the outcome in the child. And I want to stress, and I'll come to this a little later again, that the protective blood-brain barrier does not develop until the second trimester of pregnancy. So we have a double whammy now. Anything that triggers the mast cells in utero can affect the brain because there's no blood-brain barrier yet. And many of the molecules, as you will see later, that are released from mast cells can disrupt the blood-brain barrier. So whatever happens during gestation is very critical about what will happen to the newborn baby. So I wrote a review recently about mast cells in the autonomic nervous system, potential role in dysautonomia and neuroinflammation. And I made this paper available to the organizers who mentioned earlier that we'll make it available to you. It might already be in your chat, because it talks about a number of diseases other than allergies: chronic fatigue syndrome, fibromyalgia, long COVID, and autism, with numerous publications, especially for autism, about 40 publications that are relevant, and I will only cover a few. But there is now increasing evidence that the mast cells are involved in ehlers-danlos syndrome, as well as postural tachycardia syndrome, or POTS. And that's why sometimes now they call it the triad of having mast cell activation. In my mind, not necessarily mast cell activation syndrome. It can be just mast cell activation, EDS, and POTS. So what is the evidence that stress can worsen mast cells? Here are just a couple of papers, even though there are many more. This is a paper we published many years ago, where a lady had very few spots of cutaneous mastocytosis. She underwent a horrible psychological stressor. And as you can see in the picture, now she's covered UP, urticaria pigmentosa, and when the biopsies were actually stained for mast cells, you can see the black spots were mast cells. In another patient, we had indolent systemic mastocytosis, but she was invariably worsening by stress. We measured the first hormone that I mentioned under stress, corticotropic releasing hormone, and it was very high in the blood. And when we looked actually at the mast cells in her bone marrow, so the right-hand side is for the bone marrow, and we stained them for receptors for CRH, you can see in the insert those white line around the cell. So this is fluorescent antibody that recognize receptors for CRH on the surface of the mast cell. And in the bottom paper, we measured CRH in individuals that had psoriasis or atopic dermatitis. You can see CRH in controls was all the way to the bottom, was higher in psoriasis, even higher in psoriasis with higher severity, and even higher in atopic dermatitis. Now, this is critical for two reasons. Number one, because it's not only your mind. We can measure the primary hormone under stress in the blood and show that it's actually involved. And number two, we always taught in medical school that CRH is released only in the brain. Well, now you can see that it's released actually in the blood, and other colleagues that we have shown that it's released actually in other organs, such as the bladder, in a condition called interstitial cystitis. So how about the brain now? So are mast cells present in the brain? Well, we were not the first to show that. But I wrote a review back in 1990 where I called the mast cells the immune gate to the brain. Why? If you look at the middle panels at the left, this is a caricature showing the blue cells are the pericyte, the violet cells are the endothelial cells. They make up the blood-brain barrier. And you see at the top, a mast cell. Now, if you look at the right-hand side, it's a unique picture. It's 300,000 diameters magnification from our laboratory, where you can see the lumen of a blood vessel from the hypothalamus of the brain, a red blood cell inside the lumen. The E is the endothelial cell, the P is the pericyte, and now you can see at 11 o'clock a mast cell hugging the blood-brain barrier. And this is critical because, as you will see shortly, corticotropic releasing hormone stimulates mast cells and they increase the permeability of the blood-brain barrier. And on the right-hand side, you see one of many papers where the blood-brain barrier and the intestinal epithelial barrier are actually broken down in autism. And we will talk about this shortly. So where are the mast cells therefore in the brain? They're found in the meninges, the membranes that cover the brain, and quite uniquely in the hypothalamus that regulates homeostasis, and in the amygdala that regulates behavior. And as you will see shortly, the amygdala is the center for fear. So how do we prove that? Well, you can put an indicator called technetium gluceptate, which we use actually to image the brain in humans as well, in the tail vein of a mouse. You can put the mouse in a plexiglass immobilizer, and within 30 minutes, this indicator, technetium, has left the circulation and got into the brain. And if you look at the graph at the right-hand side, the plus are normal mice. Most of this indicator has left the blood and got into the brain. But if we use WW, mice which are mast cell deficient, nothing gets into the brain. So therefore, we know that CRH connect through brain mast cells to disrupt the blood-brain barrier. And to make things worse, the substance B molecule that we covered a short while ago can make the mast cell express even more receptors for CR8. So as you can see again, the mast cell is very dynamic, and one of its triggers trigger the mast cell to express more receptors for the other. We actually went ahead and used fluorescent antibodies, as I mentioned earlier, against the receptor for CR8 on the surface of the mast cell. And you can see these are human culture mast cells lighting up like light bulbs. So when you're stressed, you release CR8, then binds to the mast cells. And what happens? Well, we showed that either alone or together with another molecule released from nerves called neurotensin, the mast cells release vascular endothelial growth factor, which makes blood vessels leaky. And not only substance B can make the mast cells grow receptors for CR8, but now neurotensin does that too. So again, the mast cell releases molecules, stress releases molecules, they act on each other, and they make the mast cells now much, much more responsive. So do we know if this translates to anything in humans as it refers to the brain? Well, at the top you see allergic diseases in preschoolers are associated with psychological and behavioral problems. And you can see papers where there are neuropsychological features in mastocytosis, mastocytosis patients and cognitive dysfunction, and a paper very recent where if there was mast cell deficiency, cognition in mice was extremely improved. So there are many papers that indicate that in the brain, the mast cells are associated with neuropsychiatric problems. And if the blood-brain barrier is disrupted, substances from the peripheral blood might get also into the brain and contribute to this process. So let's take another deep breath and now try to relate what I've said so far as it relates to autism. We know, unfortunately, that 1 in 36 children in the United States are now diagnosed with autism. And papers predict that this number will become in 1 about 25 in 2025. Now, the definition of the diagnosis of autism, as you know, is impaired social communication and repetitive and stereotypical behavior. However, both I and my colleagues have noted that about 80% of the children are also hyperactive, yet hyperactivity is not part of the diagnosis of autism. And if you look at the left-hand side at the bottom, you see autism medical comorbidities. I mean, everything is medical, but in this instance, other than autism. And as you can see, there are allergies, there are food intolerance, the GI problems, hyperactivity, mitochondrial dysfunction, seizures. This makes it very difficult to study autism because we do not have a homogeneous population. And probably that's why also we still do not have a distinct pathogenesis. We lack effective treatments. And of course, the cost is enormous. Now, we will talk about a number of conditions that might contribute to autism. But over and over again, there are publications that you can see at the bottom right for preterm or early term birth and very low birth weight and separation from the mother by putting the child in an incubator is a major potential risk factor. So we wrote a review many years ago in a British journal, you can see this at the upper left-hand side, talking about various conditions that might contribute to autism. And reminding you what I said earlier, that anything that the mother is exposed to, as you can see on the cover on the right-hand side, can affect the baby in the uterus. But many publications have shown correlations, not necessarily cause and effect, but very strong correlations between allergies, asthma, atopic dermatitis. We'll talk about COVID if we have time, infections, mast cell activation, etc. A very good review was published some years back again in the journal Science about maternal immune activation, implications about neuropsychiatric disorders in general. And then papers talking about maternal either allergic problems, asthma, autoimmune diseases and the risk for autism. So the associations have already been published in large studies. Now, we published a paper many years ago, back in 2009, where we showed for the first time that children that had cutaneous mastocytosis, and you see three pictures, where the cutaneous mastocytosis is much different than a nevus. The nevus is usually bluish and does not itch when you scratch it. These are more red and they itch when you scratch them. Those children had 10 times higher chance of being identified with autism than the general population. Since then, there have been many epidemiological studies showing, as you can see, co-occurrence of autism and asthma and allergies, immune-mediated conditions, comorbidity of atopic disorders and atopic dermatitis and autism, and at the bottom right is atopy in early childhood risk factor. And there clearly, as I said earlier, about 30 to 40 publications talking about this. Yet I'm amazed that no pediatrician or even your developmental pediatrician that I've spoken to is aware of these findings or pays attention to any of this. So here are some most recent reviews about atopic diseases and inflammation of the brain and autism. Phenotypic overlap, that means that the presentation is similar between atopic dermatitis and autism. Early childhood allergy contributing. Association, again, between atopic dermatitis and neurodevelopmental disabilities. Eczema, atopic diseases, and autism. And at the bottom right, prenatal allergic inflammation, this time in rats, not in humans, actually affected what is called their dendritic spine patterning. That's a fancy way to saying neuronal communication. And as I said earlier, food allergy and food intolerance is a little different than sometimes full-blown allergy to pollen, etc. But here is a series of publications about food allergy and autism. One of the first ones, autism spectrum disorders, concurrent clinical disorders, had shown there was a very strong association. The more decimals you have for the P means probability, the stronger the association. In the Journal of the American Medical Association, Association of Food Allergy and Other Allergic Conditions, Association of Food Hypersensitivity in the European Journal of Pediatrics, and food allergy induces alteration of brain inflammation and cognitive impairments. So not only there are associations, there's actually proof, direct proof, that allergic problems lead to brain inflammation, which leads to cognitive impairment. So we were among the first to talk about focal brain inflammation. And why focal? Because we're not talking about encephalitis, we're not talking about meningitis, even though some colleagues have used such terms, which is very confusing. So now we go from the allergic issue to inflammation. So you can call it allergic inflammation if you want. So there are many papers talking about inflammation during fetal and neonatal life having worse outcomes in terms of neuropsychiatric issues. You can see in the middle panel effect of neuroinflammation on synaptic organization, again, connectivity among neurons. And at the bottom, I'm repeating what I showed you in the previous slide about dendritic spine patterning, how the neurons connect. But the mast cells in the brain are very few and in between. So we needed to see how they can actually induce brain inflammation. And in the brain, we do not have white blood cells. We have microglia. If the white blood cells get into the brain, they recognize the brain as foreign and they attack it. That's how we get multiple sclerosis. So here, a number of papers at the top, where the mast cells talk to microglia and are involved in inflammation. And very importantly, at the bottom, you can see induction of microglia activation by mediators released from mast cells, and the mast cell tryptase, which is unique for mast cells, inducing microglia activation. So there's no question that the two cells can talk to each other. Is there any evidence that microglia are activated in autism? Plenty of evidence. You can see one at the upper left-hand side among the first publications. Then they looked at the genome of the microglia was activated at the right. Again, Nature Communications, excellent journal. In the middle panel, you see now protein is involved. When the genome is activated, makes proteins, and unless you make the proteins, you don't know what is going on. And very, very important paper at the bottom in molecular psychiatry, where they took microglia from individuals with autism, they injected it actually into mice, and they caused behavior similar to what was present in humans. So you can see that we can carry, actually, there is a real pathological problem that we can carry over. And if you use SPECT scan, single positron emission computed tomography, you can actually see the activation of the microglia. And you can see at the top 3 sections of the brain from a normotypic individual, hardly any microglia are activated. And at the bottom, you see activation of the microglia. So what do we think is going on? I really believe autism is not a disease. I believe that something has happened either in utero or right after in the fear center, which is shown in red here, the amygdala. And the fear center has reset itself. So these children are just fighting for their life without realizing. That's why they cover their eyes, they cover their ears. Behavior sometimes that we consider abnormal might be very natural to them because they're trying to protect themselves. So our effort should be, can we reset this balance? Because if we reset this balance, then the brain might actually just recover on its own. And you can see here papers about prenatal stress increases the risk of autism. Again, prenatal maternal stress, ADHD and autism. And at the bottom right, prenatal stress and maternal immune dysregulation points for intervention. So it's the first paper ever that started talking about potential intervention in this instance. But how do we know what triggers the microglia? So we said the mast cell related molecules trigger microglia. So we did 2 studies back some years ago, one on the left-hand side in Greece, one on the right-hand side in the United States. We measured about 20 different peptides released from nerves to see if any was high in the blood. Now you might ask, why do we care about the blood if something is happening in the brain? Well, if a child doesn't have any other medical issue, and we measure something in the blood, it's like it's spilled over from the blood. And in fact, we found out that one molecule, neurotensin, was increased in about 50% of the cases. And if you look at the right-hand side in the other study, again, neurotensin was high. And in this case, as you can see in the title, CRH was high as well. So in at least 50% of the children, these were 4 to 12 years old, the inflammation of the brain was such that it spilled over. So what we decided to do then is take brain samples from children that died and control with normotypic children. This was published in the Proceedings of the National Academy of Sciences again. And we showed that in the microglia showing blue, in normotypic individuals, there was hardly any receptor on the surface, while in the children on the spectrum, you can see the green. There were many more receptors. So then if you look at the right-hand side, we cultured embryonic human microglia, we stimulate them with neurotensin, and we measured interleukin-1 beta. And as you can see, the microglia in the laboratory would release interleukin-1 beta. So to summarize, we feel that there is focal inflammation in the brain, especially in the amygdala and the hippocampus that carry memory. Mast cells are very close to neurons, and you can see on the left-hand side, you can see one mast cell pegging on the diagonal line, which is a neuron. The vertical line is a blood vessel. And at the right-hand side, you can see a 300,000 times magnified electron micrograph from our laboratory, where you can see some of the granules of one mast cell at the top, some granules of another mast cell at the bottom, and then is the nerve ending with the tiny little arrows indicating synaptic vesicles. But just imagine if half of each granule is this mid-tenderizer tryptase, it's going to damage this neuron ending, as will the prostaglandins and kind of other molecules. So we believe, therefore, that the mast cells activate the microglia are activated, and they damage neurons. Now, how can we now go to find out what happens to connectivity? So I showed this kind of caricature on the right-hand side where I show a spider web. So the microglia normally create the scaffolding for the neurons to crawl on. So think of them as spiders with a spider web. If, however, they're activated, they stop making the spider web, they multiply, they attack the danger or the invader, and they cause inflammation. So we have two types of damage. One, we lose the connectivity, and second, we have inflammation. We spoke about the inflammation so far. How about the connectivity? What is it that destroys the scaffolding? So we published this paper just 2 weeks ago in Molecular Psychiatry, and this and the subsequent slide will discuss this. But we looked in the brain, especially the hippocampus that carries memory, same thing also in the amygdala, for an enzyme that is called matrix metalloproteinase 9 or MMP9. And as you can see on the right-hand side, it's called the magic drug target in neuropsychiatry. And what we showed, as you can see in the graph in the middle panel, is that control mast cells and microglia in the brain were releasing a little bit of MMP9, but you can see much more in the brains of the children on the spectrum. And if you look at the color pictures on the left-hand side, you can see tryptase, double spay used with MMP9. So a mast cell in the brain of these children are positive both for tryptase and MMP9, and the right-hand side, the microglia, are also positive for MMP9 as well. So both microglia and mast cells can release MMP9. And in the middle panel on the right, you see a paper from a long time ago where it was shown that matrix metalloproteinases can be released from human mast cells. And at the bottom, you see papers where the muscles communicate with hippocampal neurons, and if the muscles are damaged, they do damage to the hippocampus. So hippocampus and amygdala are critical areas. And this is a very busy slide, but if you just focus on the bottom shaded area of the legend is the most important thing. So by looking at various genes, we showed that molecular pathways or genes that are involved in molecular pathways in extracellular matrix organization and inflammatory response were upregulated, but the connectivity was actually gone. So basically, that graph shows which genes were activated. And at the right-hand side, you can see those that were activated and those that were not activated. So the ones that were activated had to do with immunity and inflammation, and the ones that were not activated were the ones that had to do with connectivity. So again, double problem. But the mast cells also talk, unfortunately or fortunately, to other cells, such as the T lymphocytes and macrophages, and they're stimulated by toll-like receptors, and they release inflammatory molecules. And I chose to talk for the next 3 slides about mycotoxins, because I consider them a major trigger, both for mast cell-related conditions as well as for autism. So you can see effect of mycotoxins on neuropsychiatric symptoms. In the middle panel, Sporothrix, a type of fungus, can stimulate the mast cells to release only cytokines, no degranulation. And then at the bottom, a recent paper sort of about impact of mold on mast cell cytokine immune response. Is there any evidence that mold can cause neuroinflammation? Well, here's a few papers at the top, and a paper of ours that was just published a month ago. So we used, as you can see, cultured microglia, as we had done before. We stimulate them with ochratoxin, a major neurotoxin, and then we measure release of interleukin beta or interleukin 18 or IL-8, and you can see in either instance, we had actually released. So mycotoxins, which are volatile and you cannot measure them in infected houses, can actually trigger the microglia to cause neuropsychiatric problems. And there are a few papers that talk about mycotoxin exposure being related to at least increasing symptomatology related to autism, as you can see in these publications. So the next 5 slides or so, what can we do? Well, we have been studying some natural molecules for many years called flavonoids. you can see a review at the top left-hand side. And over the years, we came to the conclusion that one of them, luteolin, the structure of which is shown here, is actually probably the most efficacious in many instances. And I wrote an editorial you can see called Luteolin, the Wonder Flavonoid. It's antioxidant, anti-inflammatory, blocks mast cells, blocks microglia, and it's actually neuroprotective. And you can see at the bottom a couple of papers about the neuroprotective effects of luteolin. Now, is there any way we can block mast cells? Before, you might ask, well, what do we have any drugs for mast cells? The only drug that is available is called cromolyn sodium. But as you can see, first at the left-hand side in the middle, a paper by Dr. Church back in 1992 showed that the amount that he needed with cromolyn to block the mast cells was horrendously high, pharmacologic amounts. And if you look at the right-hand side at the top, again from Dr. Church, he showed that there's a biphasic curve. Cromolyn might inhibit a little bit and then start stimulating. And then Dr. Gali, who is the chief of pathology at Stanford University, showed that it's not even helpful in mast cells in mice. So we decided to just do a head-to-head comparison. And as you can see in this paper, International Archives of Allergy and Immunology will be published in 1 week. So I'm just showing you basically the proofs that we received. And what you can see on the right-hand side, we used exactly the same amount of cromolyn and luteolin. We pre-treated exactly for the same amount of time, 10 minutes, and then we measured a whole bunch of different molecules. You can see tryptase, histamine, cytokines, and MMP9. And as you can see, the stimulus was Ig and anti-Ig. In other words, it was a real allergic trigger. And you can see luteolin in every case, this is the third bar, inhibited significantly, while the last bar, which was cromolyn, was much, much weaker. And in some instances, like for ILA, there was absolutely no effect whatsoever. Does that mean we should not be using cromolyn? We should, especially if there's gastrointestinal problems, because cromolyn is absorbed less than 5% from the gut. It's not likely to affect anything outside the gut. But keep in mind that you can do much better, actually, by using luteolin. And in fact, we published 2 papers, one at the top and one, as you can see, in the middle, where we showed, if you look at the graph at the top at the right-hand side, luteolin was better than cromolyn, and a new molecule, methoxyluteolin, was even better in inhibiting degranulation. In the middle panel, published in the Proceedings of the National Academy of Sciences, we showed both luteolin and methoxyluteolin can block release of interleukin-1 from culture microglia almost 100%. And at the bottom, you see recent papers where they talk about luteolin as potentially immune and inflammatory modulator, and a paper at the right-hand side where luteolin actually inhibits spike protein, activating inflammatory responses in the lungs. If we have time later, we might go into that. So I helped a company called Algonaut to create some dietary supplements that contain luteolin. You can see actually at the bottom where these are available through different sites. But what I want to stress is that since we created these supplements, many other supplements have surfaced on the internet that forced me to write this editorial some years back on luteolin supplements, all the glitters is not gold. Because many supplements would say, let's say 100 milligrams luteolin, and then they'll tell you they have only 1 milligram luteolin and something else. Or you might open it and it's not yellow at all, and luteolin is yellow. So first we created Neuroprotect, that you can see on the left-hand side, that contains luteolin and quercetin, which is somewhat similar, but we wanted to add quercetin as a decoy to be destroyed quickly by the liver enzymes to allow luteolin to get into the brain. Then we realized that about 50% of people are sensitive to phenols, and both luteolin, quercetin, are phenolic, and we created pure lute that has only luteolin or no quercetin, or the Neuroprotect low phenol that has less quercetin. But many children cannot swallow the capsules, and the families had to open the capsules and squeeze the content, which does not dissolve in water. They had to put it in hummus or honey, etc. And as of about a year ago, Now there's Neuroprotect liquid in dropper bottles, and one dropperful is equivalent to one capsule. But for those who are sensitive, you can start actually with a quarter of a dropperful, etc. Now, 2 points I want to make that are very important. The one at the left-hand side, since my colleagues know that flavonoids are not absorbed more than about 10%, they will give someone 2 grams. So let's say you give someone 2,000 milligrams, they will absorb only 200 milligrams. What will happen to the rest? It stays in the gut, and as you can see on the left-hand side, it inhibits your gut microbiota. So you end up now chasing your tail because you're not absorbing anything. You might end up with SIBO. This is why we mix the luteolin and the quercetin with olive pomace oil. So after you take the olive oil, you're left actually with the seed. You squeeze that, you get a thicker type of oil that in Mediterranean countries we use it as salad dressing anyhow. But yet you can see on the right-hand side, the olive oil, whether it's virgin olive oil or pomace oil, itself has anti-inflammatory activity. So you get the benefit of Mediterranean diet, if you wish, plus higher absorption, because when they're mixed with the olive pomace oil, your absorption from the intestine becomes from about 10% to about 50%. Now, how about cognition? So most mastocytosis and mast cell activation and individuals on the spectrum would actually describe what seems to be brain fog. Sometimes we say disrupted cognition. They cannot think of the right words. They cannot multitask. The short-term memory is weaker, etc. And we developed a product called Brain Gain that, in addition to the luteolin that I've already mentioned, it contains berberine that increases blood flow to the brain. It contains biotin, contains calcium folinate, which is high octane, if you wish, folic acid, and hydroxytyrosol from olive leaves, with many, many publications that increases short-term memory. And as you can see, there are many publications in excellent journals talking about dietary flavones improving cerebral oxygenation and cognition in healthy adults, not even those that have problems. Another paper, long-term dietary flavonoid intake and suggesting cognitive decline. That was when they took it, they reversed the decline. And here's one of many papers about luteolin reducing pathologic associations in traumatic brain injury and many more in Alzheimer's. Now, before I leave this slide, I'm coming to the end. I want to indicate that, unfortunately, in many countries, supplements are not regulated. You only notify the authorities. However, in the United States, if you want to export something, you ask the Food and Drug Administration to check it as if it were a drug. They check whether it was made in good manufacturing practice facility, if the facility was FDA registered, if the amount and the purity was determined and validated by an outside laboratory. Algonaut uses urethins. And if there is at least an open label study, then they give you this certificate of free sale that you can see. It was given for Brain Gain. And as you can see at the bottom left, it expires in 2025. It's renewed every 2 years. So look for supplements that are worth your giving to your patients or your families, that are safe, that are associated with good evidence, and that they're actually contained what they say they contain. So what do we do for people that have allergic or mast cell activation problems? We start with antihistamines. Many patients don't necessarily do well with every antihistamine, so you have to try a few of them. Ropatadine, which is available in Europe but not in the United States, it's available in Canada, also block mast cells, so it's a double benefit, and it was made to block platelet activating factor. If you've got hives, you can use, of course, cortisone or a cream that I have developed that contains methoxyluteolin called Gentle Derm. If you have food intolerance on the right-hand side, you can actually use the diamine oxidase supplement. You can go up to 4 times the amount of any antihistamine, which will probably get you sedated. But before you go 4 times high, you might not give up. Might want to give a histamine 2 blocker, such as famotidine, to protect the stomach. Then you might go to liposomal luteolin and quercetin that I just mentioned are formulated so that they become liposomal. Eventually, you might move on to use an anti-leukotriene, such as montelukast. If there are problems in the brain, you might want to use an antihistamine that gets into the brain, unlike the new ones that don't, such as diphenhydramine or hydroxyzine. If you've got chronic urticaria or asthma, one might use anti-IgE antibodies to neutralize IgE, or the recent drug dupilumab that targets interleukin 4 and interleukin 13. And if there's systemic mastocytosis, now there are a number of drugs that target the tyrosine kinase in the surface that are quite effective, but not necessarily for mast cell activation. They're effective for proliferation of cells in mastocytosis. So if we're dealing with individuals that are mostly on the spectrum, and I wrote a review in Journal of Personalized Medicine about a year ago or so, clearly we have to address the hyperactivity. I tend to like ashwagandha, even though the taste is not very nice, or some drugs that are slightly anti-anxiety, such as hydroxyzine at night. If there's allergic inflammation, which I believe there is in most of the children, one can use berberine. You could use bifidobacterium in probiotics because it breaks down histamine. You can use diamine oxidase, and you can use luteolin. There are at least 2 publications, one in Greece and one in the United States, that showed that by 4 months of using luteolin, 1 capsule or the equivalent per 20-pound weight or about 10 kilogram weight, you gained, or the children gained, 1.5 a year of development, 1.5 year of development. If there's neuronal or brain fatigue, you could use folinic acid or methylfolate. And then, of course, if there are horrible behavioral symptoms, we might want to gain a little bit of time by using aripiprazole or risperidone. But those drugs just address the symptoms, not necessarily the problem. So, final slide. What are the implications? What should we kind of remember? Mast cells are ubiquitous everywhere in the body, including the brain. They contain thousands of granules that can release numerous mediators, not just histamine and tryptase, many of them having pro-inflammatory effects. There's no question that mast cells can be triggered by many allergic, neuroimmune, pathogenic, and stress stimuli. They can release molecules selectively, so we might not be able to see them. Mast cell activation occurs in many diseases, but in mast cell activation syndrome, serum tryptase has to be elevated. There's, I believe, combination of innate and external triggers that activate the mast cells in the brain as well as the microglia. And all of these molecules that are released collectively can disrupt the brain, especially during vulnerable times. So what should we do? Avoid, to the extent possible, triggers, especially mold. Keep the brain and cells healthy by giving calcium folinate or methylfolate. Reduce inflammation by using Purlut. I apologize for the spell there. Neuroprotect. Protect against brain fog by adding brain gain that has all these other molecules that I mentioned. And if we have time to talk about long COVID, we developed just recently a dietary supplement with 4 ingredients called Viral Protect. And for better or worse, I'll be giving some patents associated with brain inflammation. Okay, so the first question is about this antibody, Kangarda, recently approved in the USA for preventing immunocompromised patients with MCAS. I have not used it. I don't know the studies about it. You know, if there is immunocompromised individuals, we many times give them also just immune IgG, both intravenously as well as subcutaneously. But I will make a note of it to see if I can actually come up with more information about it. And I might be able to send something to the organizers, can then communicate. I apologize, I have not used it myself. Let me go to the next one. Garda preventive for COVID. Okay, so it wasn't for MCAS, it was for COVID. So, well, there are 2 studies ongoing now using monoclonal antibody for long COVID, but the outcome is not out there yet. I would find it hard to believe that an antibody will cross the blood-brain barrier unless it's already being disruptive. But let's wait to see what the results will be. How do we, as people with MCAS, especially those who have had neuroinflammation, were not treated but missed care? Okay, so first of all, as I said, everything that I mentioned today, especially at the beginning, was not drugs. So I would certainly try to get whichever antihistamine you can tolerate, first starting over the counter, and then you can go to some that might not be over the counter, but you definitely have to be on both H1 blocker and H2 blocker. I would definitely look for liposomal luteolin, whether it's the Neuroprotect or there's another preparation called Fibroprotect, because it will give you both quercetin and luteolin, and you will be absorbing about 50% of it. That takes time. That takes about a month or so before the mast cells are calmed down. And clearly, avoid triggers. We don't have good anti-inflammatory drugs, and we don't really give steroids, especially in children, so that would be out. But it will be very important, obviously, which is difficult, to have a health provider who kind of listens to you and is willing to kind of explore, which is one of the reasons why I left Boston after 35 years being a professor and came down to Florida, because the group that I belong to now, the Institute of Neuro-Immune Medicine, has a wonderful collection of physicians and health providers that think outside the box, and they will actually measure molecules that otherwise would not be considered by other colleagues. I would like to ask about how perimenopause severe hormonal fluctuations can affect mast cell behavior. That's an incredibly important question. We can have a whole different lecture on that. We and others have shown that estrogen augments stimulation of mast cells. Progesterone tends to be inhibitory. We've seen many times flares of various symptoms, whether it's mast cell related or migraines or interstitial cystitis or chronic fatigue, worsen either premenstrually or during ovulation, to the extent that there's no ethical or religious objection and you are of age, I would consider a progesterone birth control pill as a possibility. And there are, of course, other drugs that can put you into menopausal conditions like Lupron, but those have all kinds of other side effects. But yes, there's strong association between menstrual cycle and many of these symptoms. Can avapritinib be effective management of MCAS in whom the control inadequate? I would certainly consider it, except that insurance companies will cover it only for asthma and chronic urticaria. So if you don't have those, you might need a very smart health provider to sort of skirt the issue and find a way to have that covered. Of course, if you can pay on your own, you could certainly try it. I would have it on my list. But again, after I've tried everything else that we mentioned earlier. Can JAK inhibitors help in control of us? JAK inhibitors are a different category of inhibitors. JAK is actually a downstream event after receptors are activated. Some medications are useful for other sort of autoimmune problems. Again, before I go to a super duper drug, if I've exhausted everything, I would consider a JAK inhibitor, but they're not typically used for mast cell activation. Can mast cells be the root cause of atherosclerosis? Done genetic testing, and the main takeaway are that I have histamine overload and none of the other traditional risk factors. Absolutely, yes. We wrote actually a review many years ago, and you can find, if you put my name and heart, it will come up on pubmed.co. And it was called actually Mast Cells and Atherosclerosis and the GERD, obesity. They're important for three reasons. Number one, they line up the coronary arteries. And what is amazing is while histamine dilates blood vessels, it constricts the coronary arteries, even though they're vessels themselves. Number two, in coronary artery disease or atherosclerosis, we have the thick, solid plaque of atherosclerosis on the coronary arteries. But then the body tries to get rid of that and creates a secondary inflammatory loose plaque. It is the inflammatory loose plaque that breaks off and causes infarct. And Dr. Libby, who is chief of cardiology at Harvard in Boston, wrote a wonderful review in Scientific American many years ago, and he called it the fire within, talking about the inflammatory component. There's an additional reason, which is called the Kounis syndrome, K-O-U-N-I-S. Dr. Kounis is a compatriot of mine from Patras, Greece, and he was the first to show, and that's why now we call it the Kounis syndrome, that you can get coronary hypersensitivity due to either immunoglobulin E or other triggers of the mast cells, and molecules that are released from the mast cells can mimic a myocardial infarction, and it's not due to atherosclerosis. In addition, it's well known that stress can constrict the coronaries, even though atherosclerosis. And we already spoke about how stress can affect the mast cells. In fact, in about 4 months, the company that has made all the other formulations that I mentioned will be out with a product that will be called CardioProtect, and it will contain 4 natural ingredients that address not only the mast cells, but inflammation in the coronary arteries. So just be on the lookout for the website algonaut.com. What is the relationship, if any, of vagus nerve activity and MCAS? Excellent question. If you kindly look at the publication that I mentioned earlier, I made available to the organizers, and they put actually the link in your chat. I actually cover the vagus nerve and mast cells in that publication, so that will be a little easier. Let's just say that the vagus nerve, which is the longest nerve in the body, goes from the brain all the way to the gut, does release molecules, especially acetylcholine, that seem to affect the mast cells. And the carotid arteries have carotid bodies unrelated to the vagus nerve. But the carotid bodies, if we stroke the carotid bodies, we slow the heart down, again, because acetylcholine is released, they're loaded with mast cells. But no one has really studied them properly. Where can we find which medication affect our mast cells? Well, at least the Mastocytosis Society in the United States, which is tms.org, has actually a list of medications that can treat the mast cells. Some of them are well known, some might depend on each individual. Neuromuscular junction blockers that are used many times in anesthesia or pre-anesthetic can trigger the mast cells. Many antibiotics, especially penicillin and erythromycin-like molecules, can trigger the mast cells. Many anti-cancer drugs can trigger the mast cells. So it's kind of hard. My tryptase is okay, but my health is really bad. And as I said, tryptase, even though it's specific to mast cells, is not necessarily the main mediator involved. This is why it's important to measure the pro-inflammatory mediators as well as other mediators. For instance, in Europe, if we suspect edema or angioedema or neurogenic edema, we measure something called D-dimer, D-dimer, D-dimer. D-dimer is also very high in COVID patients because of the microthrombi, but it's involved, as I said, with edema. So that might give us some indication of what else might be going on. Sweden, me and several others in our muscle community in Sweden, an increased number of muscles in the intestine. Yes, oh my goodness, 50 to 80 is definitely intestinal mastocytosis. They look normal. It doesn't matter that they look normal because, as I showed you today, the muscle can release mediators, especially inflammatory mediators, and they look absolutely normal. So it's very misleading. If you actually go to PubMed.gov and you put my name and gut or intestine, you will see a number of papers where I talk about the importance of activation rather than the number or the appearance of the mast cells. No elevated tryptase. This is intestinal mastocytosis and not systemic. It doesn't matter. If it's intestinal mastocytosis, it has to be treated. Does this diagnosis exist elsewhere? But yes, some colleagues, especially Dr. Miner in the United States, Dr. Hamilton in Boston, talk about intestinal mastocytosis. In those cases, avoiding the foods, adding in many cases cromolyn could help. But cromolyn, unfortunately, the body gets used to cromolyn very quickly. So we usually start with 100 milligrams once a day, becomes 400 milligrams 4 times a day by 4 months, and then stops working. And it might cause explosive diarrhea as well. But I absolutely believe it has to be taken seriously and addressed. And you're not the only ones in Sweden. We have similar problems in the UK, in the United States as well, where patients just don't know where to go. So Dr. Hamilton at the Brigham and Women's Hospital is very good. I have a question. Is there increased numbers of mast cells in the gut biopsy? As I already replied, oh my God, if someone has 180 mast cells per high bar field, you have a serious problem. They don't have any clusters. Clusters, we only talk about clusters in the bone marrow, not necessarily in the gut. And as I said, I've been actually during colonoscopies looking through the colonoscope, Dr. Hamilton and others, and my colleagues don't know where to biopsy because there are no obvious lesions in the gut. So you sort of, and if you have a lesion, obviously you biopsy. Otherwise, you biopsy in a few places, especially the ileocecal junction, as I said. And what I do for anybody that I suspect intestinal mastocytosis or there is evidence, anything over 25 mast cells per high bar field will be suspicious or indicative to me. I actually ask to measure the following 3 things in the stool. Total histamine, because if histamine is released in the blood, it's broken down within a minute. If it is released in the gut, we measure it in the stool, number 1. Number 2, I always measure in the stool eosinophilic cationic protein, because eosinophils go together with the mast cells, but you don't have to have them activated visually or the numbers being high visually, because they will release eosinophilic cationic protein, and that stimulates the mast cells. And I also measure calprotectin, which I consider the best index of inflammation in the gut. If those are high, then clearly there's a problem in the gut. And if you put it together with a high number of mast cells, then you absolutely have to address it. As you know, some steroids over time can inhibit the mast cells, not immediately. That's why if we suspect anaphylaxis, we also give intravenous steroids. One steroid, budesonide, is not absorbed from the gut, and I would consider it in cases like this, along with the flavonoids that I mentioned before, because those are absorbed through the gut, so they will act in the gut as well. So measure in the stool, total histamine, eosinophilic cationic protein, calprotectin, give cromolyn up to no more than, as I said, 4 times a day, 100 milligrams, and then consider the flavonoids that I mentioned before. On your slide of different treatments, you mentioned that some of the drugs are treating symptoms, but not the root cause. I didn't catch which ones were only symptom treating. Well, by definition, antihistamines block only the effect of histamine after it's been released. Anti-leukotrienes, like Singular, block leukotriene after it's been released. We don't like to give aspirin, even though it blocks prostaglandins, because we have aspirin-induced asthma, and some individuals are sensitive to aspirin. So in terms of drugs that get to the root of the problem, if you have systemic mastocytosis or intestinal mastocytosis, the last drugs that I showed on that slide that block tyrosine kinase would be actually proper, assuming that your physician would be willing to give them to you. Otherwise, the luteolin quercetin-like supplements block the mast cells from being activated altogether. But there it's a matter of how much you can give, how much you can absorb. And I said, I never go more than 2,000 milligrams a day. And I prefer the liposomal preparations, especially the ones that I mentioned, because you get better absorption. It says, please talk about COVID. Well, let's finish the next few slides. And if we're already reaching almost 2 hours, if we have a little more time, I'll be happy to. What can we take for brain fog, cognitive issues if we're salicylate sensitive? I would not touch salicylate. And many salicylate individuals actually have so much sensitivity that are sensitive to salicylates in foods. There are many foods that contain salicylates. And as you know, aspirin is acetylsalicylic acid. So stay away from spices, spinach, things of that sort, because they contain salicylate. So my best one would be the Brain Gain, because it contains 5 different ingredients, and without necessarily trying to pitch for it, I've been taking 4 capsules of Brain Gain a day for at least 4 years. Not because I have any particular problem, because of my age, and I'm still working 10 hours a day. I just find that it's extremely helpful. Then definitely use calcium folinate or methylfolate to help with your brain cells using basically the folic acid. Folic acid has to get inside the brain cells, but then it has to be changed into its active form by an enzyme called MTHFR. About 40% of the children on the spectrum have polymorphisms or mutations of the MTHFR, so the enzyme is crawling, it just cannot make active folate. So if you give calcium folinate, you help the body at the beginning, and if you give methylfolate, you give them the end product as well. I like to give both. Unfortunately, as dietary supplements, you cannot find more than 0.8 milligrams per unit. In Brain Gain, there's 0.4 milligrams. However, with prescription, or maybe without prescription, you can get actually calcium folinate elsewhere. In Greece, there are at least 3 preparations. One is called Fedolen, that has 15 milligrams in 1 cap. There are also injectable preparations. I don't suggest the one injected, but many times for children that couldn't quite swallow, I would actually get a prescription and have them open, the family open it basically, and just use the liquid, which is just sterile liquid containing calcium folinate. All right. Thank you, Brigitte. Could you say something about not really. So what happens is when we try to use drugs that affect certain triggers, let's take IgE. If you neutralize IgE, there's no IgE binding to the mast cells. So the mast cells see that they don't have much IgE and that they downregulate the receptors on the surface. As I said earlier, some other receptors, especially the one for positively charged peptides, seem to be linked to the IgE receptor. So all of those are sort of being downregulated and destroyed. This is probably the only explanation why certain other triggers like pressure and heat are also benefited by IgE antibody, neutralizing antibodies. Is there a connection between MCAS and osteoporosis? Absolutely, yes. And again, don't stress with MCAS. It can be mast cell activation, not necessarily MCAS. And I'll make another point in a second. So the mast cell releases molecules that clearly destroy bone. Number 1, tumor necrosis factor. Number 2, something called RUNCL, but we don't measure RUNCL in clinical medicine. And number 3, which is very important, in patients that have chronic urticaria, before I would give them any drug, if you wish, I will check their parathyroid level, PTH, because both we and others have published that PTH is a very strong trigger of mast cells. And PTH, remember, takes calcium out of your bones into the circulation. So if you have high PTH, it's a double whammy. Not only you take calcium away from the bones, but you trigger the mast cell to release TNF and RUNCL, which actually causes osteoporosis. And in about 1 in 20 cases of people that had horrible problems with chronic itching and other issues associated with mast cells, I found that they had parathyroidism. These are 4 small glands sitting underneath the thyroid. And then there are drugs that can reduce the amount of parathyroid hormone released. And in certain cases, a surgeon might remove like 1 or 2 of the 4 glands, and you can still function properly. So if a patient has an allergy to apples, among many others, this quercetin is still recommended to take as a supplement. Okay, so it's a good question about anything. First of all, quercetin and luteolin are not associated with apples. The typical source Of course, many supplements don't even tell you what the source is of either quercetin or luteolin. And unfortunately, the most cheapest source of quercetin is peanut shells. And they don't tell you it's from peanut shells. So if you're allergic to peanuts, you're doomed. Another cheap source is fava beans. And about 15-20% of Mediterranean extraction individuals lack an enzyme called G6PD. And if you eat anything from fava beans, you get hemolytic anemia. So you've got to be very careful. Now, the algorithm products, the quercetin comes from the Samphora plant, and the luteolin comes from grapefruit seed extract, not the liquid. And many times we get the question, will that affect the liver enzymes? And it does not, unless with any flavonoid, you hit about 2 grams a day, in which case you will start inhibiting enzymes. And keep in mind that there are many polyphenolic compounds. Pycnogenol is phenolic, curcumin is phenolic. So don't even think of just quercetin or luteolin. Think about what other product you're taking and then get them all up to avoid any problems. Many supplements say soy flavonoids or bioflavonoids. Again, all of these flavonoids would be phenolic. So be careful to add them all up and don't exceed about 2 grams a day. I read a great reference to your work in the past. It seems MCAS is quite unknown in the Czech Republic. That is true. From my show's exam of many specialists were both individual symptoms, diagnosis, clinical psychologist, etc. I'm sorry that you had to go through so many. Would you recommend to approach for more complex assessment, middle observation, done, consultation, etc.? Okay, so without necessarily sticking my neck out, I do actually consult with patients, especially from Europe, through a different site. I can add it actually to the chat. So many of the things that I would actually want you to do, I can expand on over and beyond what I just mentioned. And there are 2 colleagues in the Czech Republic that I actually collaborate with as well. Okay, so you can see it in the chart. It's Dr. Theoharides Cyprus, the country Cyprus. The reason being is I have a nonprofit medical company in Cyprus, and it allows me to organize basically either Zoom or Skype calls, and I can direct them to specific laboratories to do the work. And I use it only for patients. So the Gmail that I had on my first slide is mostly for like a quick question or referral or so. But if you really want any more help, I would recommend that you try Dr. Theoharides Cyprus, all one word, at gmail.com, and an associate of mine will see the email, will send you some forms to fill, and then we can take it from there. What is the connection between NOD2 autoinflammatory disease and mast cells over-activation, etc.? Okay, I do not have an answer to that one. It's a good question. So NOD2 autoinflammatory diseases are recognized autoinflammatory diseases, and they actually use the term autoinflammatory, even though I think that even in what we discuss, we're talking about only inflammation. So far, I don't know about direct link. I apologize. What is the best treatment for chemical sensitivity? Again, you have to go through pretty much what I described earlier. Chemical sensitivity could, in a way, be mast cell activation unspecified, because we're talking about individuals who are very sensitive to chemicals. For instance, I can tell you, because I've traveled with one individual that had indolent systemic mastocytosis, and as you know, if you travel to another country, the moment you go through customs, they force you to go through duty-free. And this individual will just smell the perfumes and basically will collapse, or will go into a psychotic rage, and she used to call it mastocytosis. So mast cell activation could lead to many symptoms typical of chemical sensitivity, and I tend to think of them as interwoven. So I would address chemical sensitivity in the same way that I addressed so far for mast cell activation. What is the, yeah, I'm sorry. Do you maybe have an idea where I can find a list of doctors in the Netherlands? My goodness, there really aren't many, Fanny. I only know one deals with mastocytosis, but doesn't really deal with mast cell activation. And in fact, there will be a news article published soon, either in Denmark or in the Netherlands or both, about the plight of patients who are not actually taken seriously by the system. If you send me an email, I'll try to send you the one I know, but I wouldn't have very high hopes as to their addressing mast cell activation. Okay, I think that was the end of the questions. If we have another 15 minutes, I can either go through the few slides on COVID or just let them be. It's your call. What would you like? So first, there is a lot of evidence that in COVID-19 patients, there is inflammation, especially in the lungs, and there are microthrombosis. And you can see at the top papers that I and my colleagues wrote talking about microthrombosis, platelet activating factor, etc. And at the right-hand side, in the upper middle, a very important paper that histamine potentiates spike protein entry into endothelial cells. Now, I'm stressing spike protein entry because, as you will see in the next few slides, you will not find any evidence that coronavirus multiplies in the brain. So more and more evidence indicates, as you will see, that it is the spike protein that is shed from the coronavirus or is produced in the brain and affects the brain. So since histamine is primarily made, if not exclusively from mast cells, this is a very important finding. Then you can see at the left bottom a nice review, among others, about cytokine storm and functional role of mast cells. And as I showed in the caricature in the middle, the mast cells in the lungs can release interleukin 1, interleukin 6, cause inflammation. Platelet activating factor will cause microthrombosis. Therefore, it can cause edema, inflammation, and clots. And the two papers shown at the right-hand side at the bottom, there were different papers where they looked at mast cells in the lungs of patients who died with COVID-19, and they showed that the mast cells were activated in the lungs. A few papers went further. The paper at the left-hand side infected mice with coronavirus, and they looked at the lungs. And you can see at the upper right-hand side, left-hand side, the mast cells in the lungs shown violet or intact, but the ones in the mice that were infected, shown by the red arrows, are actually degranulated. Then if you look at the top graph, they measured chymase, an enzyme that is released from mast cells like tryptase, and they found the enzyme high in the serum of these patients, of the mice. And if you look at the bottom graph, they measure chymase in the blood of normal individuals all the way to the ones that were intubated. And the more severe the symptoms, the higher the amount of chymase in the blood. And what is interesting is chymase is angiotensin converting enzyme. So it might actually be related to the ACE2 receptor, which is the main receptors to which the coronavirus binds. In the paper in the top right, they measured not only chymase, but eosinophil activation that I was mentioning during the Q&A. And you can see both chymase and tryptase was much higher in the patients than in the controls. At the left-hand side, there is another paper where they triggered human mast cells and they showed degranulation with coronavirus. And I wrote a review talking about the potential association of mast cells with coronavirus. But more importantly, that links to everything we've discussed today. At the left-hand side, you see a short paper we published in the New England Journal of Medicine back in May, about a year ago, where we showed that when we actually stimulated human cultured mast cells with substance P and interleukin 33, which I had shown you before, has an augmenting effect on the release of interleukin-1. You can see it here. When we actually added recombinant spike protein, we got even more release of interleukin-1 beta. And then, if you look at the right-hand side, we investigated this further, and we decided to block the receptors for spike protein, both the ACE receptor as well as Toll-like receptors that are activated by pathogens. And as you can see on the vertical red rectangle, only when we blocked when we blocked TLR4, we blocked the release of interleukin-1 beta. And you can see at the bottom left a paper where when TLR4 is activated by the spike protein, it makes the cells create even more ACE2 receptor to which the coronavirus binds. And in the bottom middle panel, you see spike protein inducing TLR4 mediated long-term cognitive dysfunction, in this case in mice. So therefore, we should not be focusing only on the well-known ACE2 receptor, activation of TLR4 receptors, because those are the ones that release the inflammatory molecules that do the damage not only in the lungs, but in the brain as well. And in fact, there's a lot of evidence of neurologic manifestations in COVID patients, as you can see. And you can see at the left-hand side at the bottom a review about mast cell activation in long COVID. And an excellent recent paper where they showed that blood barrier disruption and systemic inflammation is associated with cognitive impairment in long COVID patients. In other words, everything we've been discussing for mastocytosis or mast cell activation patients and patients on the autism spectrum can hold true in this case. And again, activation in the amygdala by the coronavirus who gets in and sheds the spike protein can lead to activation of the microglia. To take it a step further, about a month ago or so, we published a paper where in this case we cultured microglia, not mast cells, and we stimulate them with recombinant spike protein. And again, you can see the microglia released interleukin-1, interleukin-8, TNF, and a protein called S100 beta that indicates brain damage. And if you look at the bottom left, when we blocked the TLR receptors, again, blockade of only TLR4 limited actually this response. And because of that, we decided to look at where exactly the spike protein activates. If you look at the caricature at the left, this is the last slide, the left-hand side, you can see that the spike protein can activate the ACE2 receptor, through which it is cleaved by a serine protease, and then it goes into the nucleus and produces new variants. But it can also activate mammalian target of rapamycin, which is involved in the production of inflammatory cytokines. And that activation of TLR can activate the inflammasome that can also make inflammatory cytokines. So we decided to choose molecules that can block as many of these receptors as possible. So we showed, for instance, that eriodictyol is very similar to luteolin, but it's much better water soluble. And as you can see, the first publication talks about eriodictyol suppressing cellular entry of the coronavirus. Then we used hydroxytyrosol from olive leaves that I mentioned earlier, is anti-inflammatory, as well as another component, oleuropein, in the middle panel, also from olive leaves. And as you can see, it's a potent compound against neurological complications linked to COVID-19. And we added sulforaphane from broccoli. You can see at the bottom, sulforaphane exhibits antiviral activity. And as you know, in pharmacology, the more targets you block, the more likely you're going to have efficacy, especially sustained efficacy. And in one instance, we used these in a patient who had been diagnosed with COVID-19, and her lungs were so damaged that she was put on a double transplant list in a major hospital in Tampa, Florida. And after we introduced this intervention, in 3 months, she was actually taken off the list and is doing very well, except for mild brain fog, for which she needs to continue taking the supplement. So this supplement is called Viral Protect. As I said, it contains 4 natural ingredients, each one of those with very good scientific publications of blocking coronavirus at various target points. And as I mentioned earlier about brain gain, that also has a certificate from the Food and Drug Administration called Certificate of Free Sale, renewable every 2 years. So there is hope as long as we actually act fairly quickly and sustain for at least 3 months, because the efficacy does not show up overnight. Many patients that have chronic pain or chronic itching do quite well with what we call LDN, or low-dose naltrexone. So naltrexone is an opioid receptor blocker, but a very high concentration. And my colleague and others from New York, Dr. Marcus, published a paper recently. I will send it actually, but if you look under my name on PubMed.com, it's the most recent publication, or second most recent. And we showed convincingly that unlike using a fixed dose of LDN, which is usually 4 milligrams, you have to start at 0.1 milligram and slowly titrate it up to 4 milligrams, because many get benefit before you reach at 4 milligrams. And if you push after 4 milligrams, you start getting side effects. It's again a biphasic kind of curve. I'm sorry that I didn't mention it earlier, but I'll send you the publication. You can make it available.
Just for clarification, this answer was relevant for, you said, chronic pain or chronic itching?
And chronic itching, for both.
Yeah, got it.
We use it for fibromyalgia. We use it for chronic pain, chronic itching, as I said, especially where antihistamines and others seem to fail. Reduce stress. Do anything you can to reduce stress.